Hello everyone, my name is Michael Edbert Suryanto, currently a PhD candidate in Department of Chemistry in Chung Yuan Christain University, Taiwan. In this opportunity, I'd like to share my recent research study. This study has been published in Ecotoxicology and Environmental Safety with title Using Crayfish Behavior Assay as a Simple and Sensitive Model to Evaluate Potential Adverse Effect of Water Pollution, emphasis on antidepressants. So first of all, let me introduce the background of this study. In here, we select freshwater crayfish as animal model. Why? Because through the aquatic environment, freshwater crayfish are widespread and considered an important species. Scientists also have confirmed that crayfish are suitable animal models for a variety of disciplines, including genetics, development, and cell biology. One of the freshwater crayfish species, Procambarus clarkii, or known as a red swamp crayfish, has been widely used for assessing contaminant effects on due to its biological characteristic and ecological and economic importance. This animal model has complex behavioral traits and it's considered sensitive to environmental changes, making it an excellent candidate to study psychoactive drug based on a behavioral approach. From previous studies, numerous behavioral approaches have been applied to this animal model. For example, the Felis Beatrice et al. Have evaluated cocaine and cocaine's metabolite benzoylecgonine effects on crayfish by evaluating their response toward external stimuli, feeding activity, and also exploratory behavior. Another study by Hussein et al. Also study investigate the effect of mixture pharmaceutical compound on crayfish based on their distance move, velocity, activity movement, and also time spent outside of the shelter. Meanwhile, another study by Kuo Wei et al. Also studying crayfish behavior based on their burrowing behavior, and they check the morphology, depth, and volume of the burrowing after the crayfish exposed to the methamphetamine and tramadol. However, regarding this behavioral assays, most studies only use single crayfish, and tracker software they use only retrieve the centroid coordinates of the tracker pixel matrix to locate the position of the crayfish. Some studies also conduct the behavior evaluation in multiple crayfish, but they analyze it using an ectogram, which is a manual observation that require manual checking and observation from the examiner, which is very time consuming. So for example, they will count and they will measure the certain behavior, for example, like approach with a thread without a thread display, approach with thread display, and then active claw, and then unrestrained fighting, and then they will scoring their behavior, and it will totally, they will count and measure and total it and summarize it. But this is very time consuming and require a whole length duration to watch the videos, so it's very time consuming and labor intensive. So because of that, in this study we want to apply a deep learning approach to develop an automatic crayfish behavior analysis tool. So we applied deep learning tool, which is the DeepLabCut DLC, to label multiple crayfish body part, which used to estimate and measure crayfish locomotion behavior. And then we label several crayfish body part, and then we validate and establish the method using two different approach, which is we test it on the male and female crayfish, and then also we expose it to the antidepressant sertraline. So by validating this method using different gender and emerging contaminants at antidepressant exposure, multiple behavior endpoints were applied, and then further emphasized crayfish as an excellent organism for aquatic environmental research, which is useful to be applied in the ecotoxicity study. So the experimental of this study, we put two crayfish in the water tank and then we record it using a camera from top view, and then the recorded videos then used for the training process in the DeepLabCut. And then there are 7 body parts that we label. After the video labeled, it will be marked by a circle dot with different colors, and then the output video will generate it with animal identity colored to differentiate between individuals. And then with multiple crayfish body part labeled, the distance of certain body part between individual will be measured, and then several locomotors such as behavior endpoint, distance, speed, interaction, and other can be quantified. So this is the example of programmer's Clarke body parts that being labeled. There are total 7 body parts being labeled. First is rostrum, which and then rostrum is the mouth part, and then head, talson, which is the tail, and then left claw, left salipad, right claw, and right salipad. So two crayfish body part will be labeled with the 7 body part, and then this is the video result trained with ResNet 152 models with 200,000 iteration, and the total video we use is 50 videos. With each video we use 20 frames, so total we have 1000 frames images that have been trained using the ResNet 152. So as we can see, the label DeepLabCut can uh, give a good result of tracking and labeling the crayfish body parts. After that, we calculate the distance between the left and right salipad, our right and left claw, to count the widening and narrowing of crayfish claw, which is useful to determine the aggressive behavior. For example, from this image that conducted by the from previous study, their normal behavior, they will maintain the position of their left and right claw in the uh, normal condition, but in aggressive, usually they will widening their claw. So this distance between left and right salipad we can count to we can get to count their average position, widening and narrowing, which is useful to determine their aggressive behavior. So after that, we also calculate the interdistance calculation between two crayfish, which using the same formula, the distance formula from x and y coordinate that we get from the each body part from the DeepLabCut. So we use 3 centimeter as a minimum index of interaction count because the average distance from claw to rostrum approximately around 3 centimeter. So this distance between crayfish lower than 3 centimeter will be counted as a approaching or interaction. Next, we another behavior endpoints such as fighting and chasing, also calculated using the same approach of distance calculation, but the body parts that we use for the distance calculation is different. For fighting, we put some simple formula calculation using if formula. So if the left claw crayfish 1 and right claw crayfish 2 is in the close proximity, and also the right claw crayfish 1 is in close proximity to left claw crayfish 2, for example like this image, it will count as a fighting. And next one is the chasing. Chasing were measured using the distance between rostrum, which is the mouth part of the crayfish, and the talson, the tail part of the other crayfish. So it will count as a chasing if they have maintained in close proximity distance. So in this slide, this is a several representative images of crayfish behavior which have been evaluated in this study. For example, the outspread claw. As we can see, this when they are widening their claw, it will count as aggressive behavior. And then crayfish also display their aggressive behavior by show agonistic behavior when they brace themselves against their opponents. And then in the figure C, crayfish will chase their opponent because in this scenario, one crayfish would have a winning experience and it will emerge as a dominant individual who usually will chase off their opponents. And then all these endpoints were quantified using the distance between each specific body part. And there are, for this table, there are total 13 behavior endpoints used in the study, such as total distance, speed, freezing time activity, normal movement, rapid movement, thigmotasis, intercrayfish interaction time, and also intercrayfish longest time interaction, fighting time, longest fighting time, chasing time, longest chasing time, and claw exchange ratio. So in this slide, this is the example result of the label and track result from DeepLabCut, which will be output and saved into CSV file that contain X and Y coordinate from each body part. So all 7 body part will save from the rostrum, head, tailson, left pincer, left salipad, right pincer, and right salipad from 2 individual crayfish. So for example, this is the crayfish 1, and from another row we'll save our crayfish number 2. And this position that we use to calculate the distance formula between each body part, and then we will use this to measure the behavior endpoints. So because if we use only a regular traditional Excel calculation, it will be very time consuming. So in this study, we also developed a crayfish behavior analysis app tool, which can be downloaded from this GitHub link domain. So we just need to install and run it using Anaconda prompt. So this is the graphical user interface of our app. So we built this app for an automatic data processing to extract behavioral endpoints from the trajectory result, which I already showed from the previous slide. So this tool have GUI to help for analysis, and also we can set the parameters along with the conversion rate and then the center X and Y position, also the FPS of the video and the duration of the video, and also the threshold, for example, like the position, minimum position that previously we already set at 3 centimeter, and meanwhile same with the other else. We can set this to help for us so we can quantify the agonistic behavior in crayfish, replacing the manual observation scoring system. And this is the sneak peek of our apps. And in here we can select which endpoint that want to be saved. For example, like the pincer stat, which is the distance widening and narrowing claw, and then movement stat, interaction stat, fighting stat, and chasing stats. So this will output the result, and it will be saved, and we can just export it to Excel file, and also can draw the graph. So the graphical distance position will indicate the up and down of the crayfish movement. So for example, this is the distance between left and right pincer of crayfish 1. So the higher distance of left and right pincer mean indicate they are in the showing an aggressive behavior. Similar in crayfish number 2, they also can display an aggressive behavior based on the distance between left and right pincer. So with this app, we can also do the batch processing analysis. So we can import several multiple CSV files for batch processing, and all the files will be calculated at once. So it will help us to finish our calculation quickly. And The analysis process will be running in the Anaconda prompt, and the summary will be exported to based on the output folder, and it will automatically process, and the summary file will be saved in that folder. So it only takes 1 minute to finish the batch analysis. For example, in this case, we use 10 Excel files. So compared to the manual VBA or traditional Excel analysis, it will take around maybe 1 minute to analyze single Excel file. So by using the Crayfish behavior analysis tool, this tool we can speed up the analysis process 10 times faster than the traditional one. So this is the result of our study. In here, we compare by using the develop method. We compare the locomotor activity between females and males. As we can see, the left one is the female, meanwhile the right one is the male crayfish. So this is female and female. This one is male and male. As we can see from the video, male crayfish tend to have a fighting behavior. As we can see, they tend to brace their opponent, to pinch or maybe to push away their opponent. Meanwhile, in the female, they just regularly exploring the water tank. So based on the result, the male group show a significant higher claw extent ratio than females, and then claw extent from the body indicate aggressive action from crayfish. Meanwhile, the female group displays significant higher total distance move and average speed, as also seen from the video, because female interacted less with their conspecific, resulting in higher exploration activity. Meanwhile, male tend to stop moving because they tend to fight in quite long time with their conspecific. And this also can happen because male crayfish tend to be more alert when introduced to another male, which creates competition for territory control. And regarding the social interaction, male crayfish also display significant higher and longer interaction than female. So compared to male crayfish, female crayfish only approach the other female in short amount of time, followed by no response or ignored by the conspecific. And in this result, it display a male crayfish fighting behavior and female crayfish fighting behavior. As a result of this, only male that display active agonistic behavior, which male to male crayfish will approach with threat display using an open claws to grab and fight the opponents, as can be seen from this duration event that show a tight close gap distance from these 5 events 1, 2, 3, 4, and 5. And for a relative long period of time, the close distance between male crayfish was maintained in proximity, as displayed in this figure. But in the contrary, the females did not show the same interaction as male group. Female crayfish doesn't show a maintained position of their claw to each other, unlike the male crayfish. So in addition, from all videos recorded, none of the female crayfish display aggressive and fighting behavior. So we can conclude that fighting behavior is only observed in the male crayfish. Thus, therefore, using male crayfish was preferable for behavior study involving interaction. So we conducted the behavioral test to evaluate the anti-depressant effect in pair of male crayfish. So we divided into 5 different groups: control to control crayfish pair with control crayfish, and then sertraline 7 days exposure pair with control crayfish, and then 14 days sertraline exposed crayfish pair with control, and another group sertraline 7 days sertraline exposed crayfish pair with another 7 days sertraline exposed crayfish, and the last group is 14 days sertraline treated crayfish paired with 14 days sertraline treated crayfish. So based on this result, we can conclude after exposure with sertraline either 1 ppb sertraline for 7 days or 14 days, there are several different of crayfish behavior. The the clear result can be shown by the interaction time. So almost all the crayfish that exposed to the sertraline will have less interaction time, and also some of the alteration behavior also observed in the sertraline 14 days group, which displayed by the significant increase in the extend claw, total distance moved, average speed, and also the rapid movement. Either they are both to control or another sertraline exposed crayfish. So based on this, crayfish exposed to sertraline show hyperactivity and aggression. And however, despite of they show hyperactivity and aggression, the sertraline also cause crayfish to be less attracted and interact with their conspecific. And then we also evaluate their fighting behavior. Based on the fighting behavior, sertraline can reduce the fighting time percentage. The agonistic behavior of fighting and chasing was evaluated after pairing these two crayfish. So either exposed to sertraline or the sertraline group pair with the control in the fighting arena. So in here, the total fighting time of sertraline 40 days exposed, it have less fighting percentage because it's plausible because the sertraline treated group have more aggressive behaviors, so it will already determine who's the winner, who's more dominant. So the control crayfish tend to be escaped from the sertraline treated crayfish. So from this, the dominant crayfish frequently chase the conspecific by lifting the salipad, but the tracing interaction for the sertraline group is lower than the control. In addition, the longest chasing time from all sertraline treated group also display no significant difference. Yeah, because the sertraline treated group already have a dominant behavior, they have poor bold behavior, so they already maintain their distance, so they already become the winner. So the control will tend to retreat from the dominant crayfish. So we also evaluate the behavior alteration in crayfish exposed to different antidepressant by using principal component analysis and heat map clustering to see whether the effects of this drug show any similarities. So in here, it shows that all the antidepressant exposure treatment have distinct relative to the control as displayed by the PCA graph. So the 14 days exposure, which is indicated by the blue and purple color, have distinct relative to the control. And further detail based on the clustering map revealed that the sertraline 40 days exposure have the farthest relation compared to the control. As you can see, there are two major groups. The left one left with the sertraline 14 days group. So it showed that the disruption is caused by the antidepressant effect of this animal, further disrupted by the 14 days exposure. So in this study, we have pioneered an innovative automated approach for analyzing crayfish behavior using a trained DLC model, DeepLabCut model with the ResNet 152 network. And our method not only retrieve precise X and Y coordinate position, but also combined with Python-based script to extract a spectrum behavioral endpoints. This automation also offers significant advantages over traditional manual observation, including real-time labeling and enhanced accuracy in quantification. And based on the validation method, our results also show that male crayfish exhibit more aggressive behavior than the female. In addition, also 14 days exposure of sertraline resulted in increased locomotion activity and aggressive tendency, but reduce the social interaction in freshwater crayfish. So, with the increasing number of water pollutants, aquatic animals such as fresh crayfish for crayfish play a vital role in assessing ecological impacts. So, overall, this method present new technology, new method, and automated behavioral assessment that provide robust tool for ecotoxicology studies using crayfish as a model. And that brings us to the end. I'd like to thank you for your time and your attention. If you have any question and need further information, feel free to contact me at my email and Facebook. Thank you and goodbye.